Dictyothiodspira, a Heliodopspira, predominant organic matter originating from primarily reproduction by oxygenic or anoxygenic phototropes, then sets the stage for development of the extreme halophilic archaea, all of which are chemoorganotrophic. In addition, a few extreme halophilic anaerobic chemoorganotrophic bacteria such as Halioanaerobium and Haliobacterioids thrive in such environments. Marine salterns also habitats for extremely halophilic prokaryotes. Marine salterns are small enclosed basins filled with seawater that are left to evaporate, yielding solar sea salt. As salterns approach the minimum salinity limits for extreme halophiles, the waters turn a reddish-purple color due to the massive growth called a bloom. A halophilic archaea, the red coloration apparent, and C comes from carotenoids and other pigments discussed later. Morphologically unusual prokaryotes are often present in salterns, including even square bacteria. Extreme halophiles have also been found in high salt foods such as certain sausages, marine fish, and salt pork. Taxonomy and physiology of extreme halophilic archaea. The current recognized species of extremely halophilic archaea, 16S ribosomal ribonucleic acid, sequencing and other criteria have defined 10 genera of extreme halophiles. The extreme halophilic archaea are frequently referred to as collectively as Haliobacteria. Because the genus of Haliobacteria was the first in the group to be described and is still best studied representative of a group, Natronobacterium, N.A. Natronomonas, and their relatives differ from the other extreme halophiles in being extremely alkalophilic, alkaliphilic, as well as halophilic, as befits their soda-like habitat, and growth of Natronobacteria is optimal at very low Mg at M2 concentrations and high pH. Extremely halophilic archaea strain gram-negatively reproduced by binary fission and do not form resting stages or spores. Most Haliobacterium are non-motile, but a few strains are weakly motile by flagellar means. The genomic organization of Haliobacteria and Haliococcus is unusual in the large plasmids containing up to 25 to 30% of total cellular DNA are present, and the GC base ratio of these plasmids, 57 to 60% GC, is significantly different from that of chromosomal DNA, 66 to 68% GC. Plasmids from extreme halophiles are among the largest naturally occurring plasmids known. Extremely halophilic archaea are chemoorganotrophs, and most species are oblique aerobes. Most Haliobacteria use amino acids or organic acids as energy sources and require a number of growth factors, mainly vitamins. For optimum growth, a few Haliobacterium species oxidize carbohydrates, but this ability is relatively rare. Electron transport chains containing cytochromes A, B, and C are present in Haliobacteria, and energy is conserved during aerobic growth, a proton motive force arising from membrane-mediated chemoosmotic events. Some Haliophilic archaea have been shown to grow anaerobically, anoxic growth at expense of sugar fermentation and by anaerobic respiration. Linked to the reduction of nitrate or fumarate has been demonstrated in certain strains. Water balance in extreme halophiles. Extreme halophilic archaea require large amounts of sodium for growth. In the case of Haliobacterium, where detailed salinity studies have been performed, the requirement for Na cannot be satisfied by replacement with another ion, even with the chemically related ion K+. We learned in section 6.12 that certain microorganisms can withstand the osmotic forces that accompany life in a high solute environment by accumulating or synthesizing organic compounds intercellularly. The lateral compounds are referred to as compatible solutes. These compounds counteract the tendency of the cell because dehydrated under conditions of high osmotic strain by placing the cell in positive water balance with the surroundings. As a compatible solute, cells of Halobacteria pump large amounts of K+ from the environment into the cytoplasm, such as the concentration of K+ inside the cell is considerably greater than the concentration of Na+ outside the cell. The Halobacterium cell wall is composed of glycoprotein and stabilized by sodium ions. Sodium ions bind to the outer surface of the Halobacterium wall and are absolutely essential for maintaining cellular integrity. When inefficient Na+ is present, the cell wall breaks apart and the cell lyses. This is a consequence of the exceptionally high content of the acidic negatively charged amino acids, aspartate and glutamate, in the glycoprotein of the Halobacterium cell wall. The negative charge is contributed by the carboxyl groups of these amino acids are shielded by Na+. When Na+ is diluted away, the negatively charged parts of the proteins activated repel each other, leading to cell lysis. Halophilic cytoplasmic components. Like cell wall proteins, cytoplasmic proteins of Halobacterium are highly acidic, but it is K+, not Na, that is required for activity. This, of course, is not surprising since K+ is the predominant internal cation in cells of Halobacterium. Besides a high acidic amino acid composition, Halobacteria cytoplasmic proteins typically contain very low levels of hydrophobic amino acids and lysine, a basic amino acid compared with those of non-haliophiles. This also makes sense since in a highly ionic cytoplasm, highly polar proteins tend to remain in solution, whereas nonpolar molecules would tend to cluster and perhaps lose activity. The ribosomes of Halobacterium also require a high K+ levels for stability. Ribosomes of non-haliophiles have no K+ requirement. It thus appears that the extremely halophilic archaea are highly adapted both internally and externally to life in a highly ionic environment. Cellular components exposed to the external environment require high Na for stability, whereas internal components require high K+, with the exception of few extremely halophilic members of the bacteria that also use K+ as a compatible solute. In no other group of prokaryotes do we find this unique requirement for such high amounts of specific cations. Bacteriorhodopsin and light-mediated ATP synthesis. Certain species of extremely halophilic archaea are capable of light-mediated synthesis of ATP that does not involve chlorophyll. Thus, it is not photosynthesis. The highly pigmented nature of extremely halophilic archaea is visible in Figure 13.2. Pigmentation is due to red and orange-colored carotenoids, primarily C50, carotenoids called bacterioruberins, and also to inducible pigments involved in energy metabolism. Under conditions of low aeration, the Halobacterium selenarium and certain other extreme halophiles synthesize and insert a protein called bacteriorhodopsin into their membranes. Bacteriorhodopsin was named because of its structural and functional similarity to the visual pigment of the eye. Rhodopsin conjugated to the bacteriorhodopsin is a molecule of retinol and carotenoid-like molecule that can absorb light and catalyze formation of protein motophores. The The retinol gives bacteriorhodopsin a pure hue. The cells of Halobacterium switch from growth under conditions of high aeration to oxygen-limiting conditions, a trigger of bacteriorhodopsin synthesis, gradually change from an orange to red color to a more reddish purple as they synthesize bacteriorhodopsin and insert it into the cytoplasmic membrane. Bacteriorhodopsin absorbs light strongly in the green region of the spectrum at about 570 nanometers. The retinol of bacteriorhodopsin, which normally exists in an all-trans configuration, becomes excited and converted to the cis form following the absorption of light. The transformation results in the translocation of protons to the outside surface of the membrane. The retinol molecule then returns to its more stable all-trans isomer in the dark along with the uptake of proton from the cytoplasm, thus completing the cycle. As protons accumulate on the outer surface of the membrane, the proton mode of force increases until the membrane is sufficiently charged to drive ATP synthesis through action of a proton translating ATP. Light-mediated ATP production of Halobacterium solanarium has been shown to support slow growth of this organism anaerobically under nutritional conditions in which other energy-generating reactions do not occur, and light has been shown to maintain the viability of anoxic cultures of Halobacterium. Incubated in the absence of organic energy sources, the light-stimulated protein pump of H. solanarium also functions to pump Na plus out of the cell by action of Na plus H plus antiport system, and to drive the uptake of a variety of nutrients, including the K plus needed for osmotic balance. The uptake of amino acids by solanarium has been shown to be indirectly driven by light, because the transport of amino acids occur with an Na plus uptake by an amino acid Na plus symporter. Continued uptake depends on the removal of Na plus via the light-driven Na plus divided H plus antiporter. Other rhodopsins. At least 3 other rhodopsins besides the bacteriorhodopsin are present in membranes of Halobacterium solanarium. Halorhodopsin is a light-driven pump that pumps chloride into the cell as the concentration for K+, the retinol of Halorhodopsin binds chloride negative and transports it into the cell. 2 sensory rhodopsins are present in H. solanarium. These are light sensors controlling phototaxis, the movement towards light, and the organism through the interaction of cascade of proteins similar to what drives chemotaxis. Sensory rhodopsins affect flagellar rotation to move cells of H. solanarium toward light, where the bacteriorhodopsin can then function to make ATP. We will learn in our discussions of marine microbiology that species of bacteria exist in open ocean waters that contain bacteriorhodopsin like proteins called proteorhodopsins.